Right now, like inside your bloodstream, billions of microscopic packages are being rapidly swapped between your cells,
Just an unimaginable number of them.
right? And they're carrying complex genetic blueprints, protein instructions, basically all this crucial information that literally dictates how your body functions.
Yeah, it's a massive, highly coordinated biological postal system.
Exactly. But here's the wild part. For like 80 years, science thought this entire system was literally just garbage.
It's, uh, it's a humbling piece of scientific history, really. If we go back to the source material, specifically to around 1946, researchers observed this strange, mysterious clotting factor in blood plasma.
But they had no clue what it actually was, right?
No idea. So they basically shrugged and described it as, quote, cellular dust.
Cellular dust.
Yeah.
Just microscopic debris flaking off our cells.
Well, welcome to our deep dive into the source material on exosomes. Our overarching mission today is to explore how these tiny packages actually work.
And more importantly, how scientists verify them. Yes, how they separate a genuine, life-saving cellular therapy from that actual biological dust. Because today, that quote-unquote dust is the foundation of cutting-edge nanomedicine. It's in high-end cosmetics, and it's used in clinical-grade, intimate exosome preparations for highly sensitive regenerative therapies.
Which is a massive industry now.
It really is. And to prove those preparations are pure, scientists rely on incredibly specific cell surface markers called CD9, CD63, and CD81. Okay, let's unpack this, because the shift in perspective here is just staggering.
Oh, absolutely. I mean, it wasn't even until the 1980s that the term exosome was formally adopted. And even then, the prevailing theory was still that cells were simply using them as, like, microscopic dumpsters.
Right, just throwing out unwanted protein.
Exactly. It took decades of improved technology to realize these aren't trash bags at all. They're the fundamental way cells communicate with each other.
Which is exactly why you, listening to this right now, need to care about the mechanics of this. Because whether you're researching non-invasive cancer diagnostics, or maybe you're considering a clinical-grade regenerative serum for intimate wellness and skincare, understanding how scientists verify the purity of these products is crucial knowledge.
Because the stakes are incredibly high.
Yeah. If we can't accurately identify the real messengers, you might just be buying a very expensive vial of actual cellular garbage.
And identifying them at that scale is, well, it's a monumental technical hurdle. We're talking about structures measured in nanometers. You can't just hold a vial up to the light.
Right. You need molecular proof.
Right.
So to figure out how we identify them, we have to start at the beginning. I know they're essentially tiny bubbles, but what actually constitutes an exosome on a physical level?
At its foundational core, an exosome is a nano-sized vesicle enclosed by a lipid bilayer.
So a fat bubble.
Yeah. To put it in everyday terms, imagine a microscopic droplet of water surrounded by a thin, flexible shell made of 2 layers of fat molecules. They're impossibly small, typically ranging from 30 to 150 nanometers in diameter.
But their size isn't the main thing that defines them, is it?
No, it's not. What separates a true exosome from just a random piece of cellular debris is how it's born.
Because they aren't just pinching off the outside of the cell membrane randomly, right?
They absolutely are not. If they did that, that would be a completely different class of vesicle, something we call a microvesicle or an ectosome.
Okay, so exos are different.
Much different. They possess a much more complex, highly regulated origin story deep inside the cell. It begins when the cell's outer membrane invaginates. It actually folds inward to swallow a tiny bit of the outside environment.
Creating an internal bubble.
Yes, exactly. We call that an early endosome.
Okay, so the cell swallows a bubble. What happens next?
Then it goes a step further. The membrane of that internal bubble starts folding inward on itself, creating even smaller bubbles inside the larger one. We call this a multivesicular body, or an MVB.
Oh, wow. So it's like a cellular Matryoshka doll. You have the main cell, which contains a bubble, and inside that bubble is a whole swarm of even tinier bubbles.
That captures the dynamic beautifully, actually. Those tiny interluminal vesicles inside the MVB, those are the future exosomes. The cell then actively loads them up with specific RNA, DNA, and proteins.
It's physically packing them with instructions.
Precisely, and the critical moment happens when that larger bubble, the MVB, travels back to the edge of the cell, fuses with the outer plasma membrane, and just bursts open.
Spilling all those tiny inner bubbles out into the body.
Right, and only once they are released into the extracellular space do we officially call them exosomes.
So if they go through this highly specific, highly regulated internal shipping process, they must pick up something along the way that identifies them. Like If I'm receiving a highly classified courier package, there's usually a barcode or a tracking label on the outside of the box, proving it went through the proper channels.
That's a great way to think about it.
So what does an exosome's ID badge actually look like?
Well, that brings us directly to the stars of the source material, a family of integral membrane proteins known as the tetraspanins. In the world of exosome research, 3 specific tetraspanins have emerged as the absolute gold standard for identification.
And those are the markers we mentioned earlier, right?
Yes, they're known as CD9, CD63, and CD81.
CD9, CD63, and CD81. Okay, so these are the barcodes stamped on the outside of the exosome. But what actually are they? The name tetraspanin kind of sounds like it's describing a physical shape.
It is entirely descriptive of their physical architecture. Tetra meaning 4, right? These proteins possess 4 transmembrane domains.
Meaning they go through the membrane 4 times.
Exactly. Rather than just sitting on top of the vesicle like a sticker, the protein physically snakes back and forth, weaving across that lipid bilayer fat bubble 4 separate times.
Well, that makes me think of a tailor stitching a thick reinforced handle onto a delicate silk bag. It's not just a patch. It's physically woven into the fabric to give it structural integrity.
A very apt comparison, because these tetraspanins are not just passive ID badges. They form what researchers call tetraspanin-enriched microdomains, or TEMs.
TEMs, got it.
And these microdomains are highly active, specialized zones on the surface of the vesicle. They organize the entire membrane architecture. They regulate how the exosome adheres to other surfaces.
So they're functional?
Very much so. They actually actively facilitate the fusion of the exosome when it finally reaches the target cell it's trying to deliver its message to.
So they aren't just an ID badge. They're the literal key card that unlocks the door to the receiving building.
That's spot on. And what the source data makes very clear is just how intentional this process is. These tetraspanins are unbelievably abundant on exosomes.
Like how abundant?
We see them enriched anywhere from 7 to 124-fold compared to their presence on the parent cell's own plasma membrane. The cell is expending vital energy to actively pack these specific functional proteins onto these vesicles before sending them out.
Which is great for the cells, but also incredibly convenient for the scientists trying to track them down.
Yeah.
But so what does this all mean for clinical applications? Because here's where we hit a massive roadblock.
The purity problem.
Right. If we're trying to manufacture a clinical-grade intimate exosome preparation, let's say a highly targeted, soothing regenerative serum for sensitive skin applications, absolute purity is non-negotiable.
Definitely.
But these things are 30 to 150 nanometers wide. You can't just look at a vat of liquid and know if it's pure. You could be looking at a multi-million-dollar therapeutic, or you could be looking at a vat of busted cell fragments that might trigger a massive immune response. So how does the scientific community actually police this?
This is a major concern, and it's where the International Society for Extracellular Vesicles stepped in to create order out of the chaos. They established a rigorous set of standards known as the MICV guidelines.
MICV.
Yes, the minimal information for studies of extracellular vesicles. These were heavily updated in 2018 and again in 2023. And the MICV guidelines explicitly dictate that to definitively prove you have a pure exosome preparation, you cannot rely on just one test.
Makes sense.
You must detect at least 3 protein markers.
And I'm assuming those are our tetraspanin ID badges, CD9, CD63, and CD81.
Usually, yes. You must definitively prove the presence of positive markers like those tetraspanins. But the guidelines also mandate a second, equally crucial step. You absolutely must test for negative markers.
Okay, hold on. I need to push back on that logic. Wait, why do we need negative markers if we already found the positive ones?
Yeah.
Like, if I walk up to a security checkpoint and scan my valid CD9 ID badge, and the light turns green, you don't also need me to provide proof that I'm not a trespasser. The positive identification should be enough, shouldn't it?
This raises an important question, and it seems counterintuitive until you look at the violent physics of how we extract these things.
Violent physics.
Oh, yeah. When scientists isolate exosomes from a cell culture, they frequently rely on a technique called ultracentrifugation.
Which is basically putting the sample into a highly advanced centrifuge and spinning it.
But not just a standard spin. We're talking about subjecting the sample to forces exceeding 100,000 times the force of gravity.
Wow.
Imagine taking a city, putting it in a blender, and spinning it at supersonic speeds just to force the absolute smallest dust particles to the very bottom of the tube. Under that immense stress, it is incredibly common for the parent cells to simply shatter.
Oh, man. So they just break apart.
Exactly. And if they die and break apart, you get all sorts of internal cellular junk mixed in with your pristine exosomes.
Oh, wow. So you aren't just looking for the courier package. You have to prove that the courier's original office building didn't violently explode and land in the same pile.
That is the exact nightmare scenario for clinical purity. Therefore, researchers must test for the absence of non-vesicular impurities. A classic negative marker they test for is calnexin.
Calnexin. What does that do?
It's a protein that functions exclusively inside the endoplasmic reticulum, which is the protein factory deep inside the parent cell. It has absolutely no business being on a secreted exosome.
So if you run a biochemical test on your high-end regenerative exosome serum and you get a positive hit for calnexin,
It guarantees your parent cells ruptured and spilled their internal guts into your sample. Your preparation is contaminated with cellular debris. It is not a pure clinical-grade product.
that makes total sense. Are there other negative markers?
Another mandatory one to check is albumin, which is an incredibly abundant plasma protein. You test for albumin to prove you've actually washed away all the background fluid and isolated nothing but the vesicles.
Okay, right. You find the positive CD markers to prove you have vesicles of endosomal origin,
Yeah.
and you use the negative markers to prove the sample is clean. But reading through the source material, here's where it gets really interesting. There is a pretty massive plot twist regarding these quote-unquote gold standard tetraspanins. For years, the scientific community treated CD9, CD63, and CD81 as universal. But they aren't actually universal at all, are they?
They are not. And discovering that was a massive paradigm shift. As our detection technologies became more sensitive, we realized the biological landscape is vastly more nuanced than we thought.
How so?
Well, the data shows that different cell lines behave entirely differently. A prime example in the literature is Jurkat cells. They're a specific type of human T lymphocyte along with certain B-cell lymphomas. They produce abundant, highly functional exosomes, but those exosomes are completely missing the CD9 marker.
Wait, completely missing. So if you were a manufacturer using a purification tool that only looked for the CD9 barcode to verify your product, you would look at that batch and conclude there are no exosomes here, and you'd just be dumping a perfectly good batch down the drain.
You would be entirely wrong, yes. And the reason for this immense heterogeneity likely stems from the physical geography of the cell.
The geography.
Yeah, these proteins originate and accumulate in different distinct zones. The research indicates that CD63 tends to localize very heavily in those deep internal endosomal compartments we discussed earlier.
Okay, and the others?
Meanwhile, CD9 is predominantly stationed at the outer plasma membrane, and CD81 just kind of seems to float between both locations.
So depending on the exact microscopic location where that tiny vesicle forms and buds off, it might grab a completely different handful of proteins to stitch into its membrane.
It goes even further than that, actually. We now have stunning visual proof of this heterogeneity thanks to single vesicle imaging techniques, specifically total internal reflection fluorescence microscopy, or TIRF.
Okay, hold on, you lost me there. A standard exosome is what, 50 nanometers wide?
Roughly, yes.
But a single photon of visible light has a wavelength of roughly 400 to 700 nanometers. It's like trying to measure a grain of sand using a yardstick. How are scientists physically using light to look at individual vesicles that are smaller than the light itself?
That is the genius of TIRF microscopy. It sidesteps the normal diffraction limit of light. Instead of shining a beam directly through the sample, which would just create a blurry mess,
Right.
TIRF fires a laser at a glass slide at a very specific steep angle. The light completely reflects off the glass, but it creates this incredibly thin electromagnetic field on the other side called an evanescent wave.
An evanescent wave.
Yes, and this wave only penetrates about 100 nanometers into the sample.
So it only illuminates the absolute thinnest possible slice right at the surface of the glass.
Precisely. So they immobilize the exosomes on that glass chip and tag the different tetraspanins with different colored fluorescent antibodies. When that evanescent wave hits them, it lights up individual single vesicles in the dark.
That's amazing. And what do they see?
When scientists observe this, the distribution is fascinating. CD9 and CD81 frequently co-localize. You will see a single vesicle light up with both of those markers. But CD63 is frequently found flying solo on its own completely distinct subpopulation of vesicles.
That is incredible. So if you look at a swarm of exosomes from the exact same parent cell, some are wearing a CD9 and CD81 uniform, and others are wearing only the CD63 uniform.
Furthermore, the TURF data shows that tetraspanin positivity overall is heavily correlated with the absolute smallest extracellular vesicles, those under 100 nanometers. As the vesicles get larger, those specific markers become drastically less frequent.
So it's less like one universal ID badge and more like different departments in a massive corporation having slightly different security badge colors. Like the marketing department might require blue and green badges, but the accounting department only uses red. Not every single courier in the building carries every single color badge.
Which perfectly illustrates why the MISEV guidelines require a comprehensive panel of markers. If you only screen for the red badge, you're going to completely miss all the marketing couriers.
Right.
By testing for a panel of all three, CD9, CD63, and CD81, you ensure rigorous verification and capture the full spectrum of exosomal subpopulations rather than relying on a single point of failure.
Okay, so we can isolate them, we understand why they wear these specific tetraspanin badges, and we can visualize them one by one in a high-tech lab. But let's scale this up. How do we take this to the clinic?
The manufacturing side.
Yeah, because verifying a handful of pristine exosomes on a glass chip is one thing, but manufacturing clinical-grade preparations, like those intimate regenerative therapies we mentioned earlier under good manufacturing practice or GMP, is a totally different beast. If I'm making a 50-gallon vat of this stuff, how on earth do I verify the entire batch?
Well, if we connect this to the bigger picture, GMP manufacturing requires a deeply multi-tiered approach to quality control. You can't just run a quick EXELISA test for those tetraspanins and call the batch safe.
Real quick, an EXELISA test. I'm assuming that's essentially a biochemical assay, like a highly targeted pregnancy test, but for exosomes.
That's a great analogy, actually. It uses specific antibodies that bind to your target proteins and produce a measurable color change or a light signal if the CD markers are present.
Okay, simple enough.
But while an EXELISA proves what proteins you have in the vat, you also have to prove how much physical product you have and what shape it's in.
Right, because if you're selling a clinical-grade therapeutic, the actual dosage of particles is everything.
Exactly. To solve the dosage problem, GMP facilities pair the biochemical marker tests with a physical measurement technique called NTA, or nanoparticle tracking analysis. NTA uses laser light scattering to track the Brownian motion of particles suspended in liquid.
Brownian motion being like the random, jittery movement of microscopic particles as they constantly collide with water molecules.
Exactly. Smaller particles jitter faster, larger ones jitter slower. By tracking that movement with a laser and a camera, a computer can calculate exactly how many particles are in the fluid and exactly how large they are.
You get an exact count.
Yes. For therapeutic or highly targeted cosmetic applications, the industry threshold often requires a massive concentration. We are talking a minimum of 3 times 10 to the 10th particles per milliliter.
30 billion particles in a single milliliter of fluid. That is staggering.
It's an astronomical number, but NTA has a blind spot. It only counts physical geometry. It cannot tell you if that 50 nanometer particle jittering around is an exosome or just a random clump of shattered protein.
Oh, right. It just sees the shape.
Exactly.
Yeah.
That is why the tetraspanin marker data and the NTA data must be evaluated together. And for ultimate clinical certainty, GMP protocols add a third visual tier: transmission electron microscopy, or TEM.
Getting actual eyes on the target.
TEM uses a beam of electrons instead of light, allowing us to see the physical structure. Genuine exosomes have a very classic, distinct cup-shaped morphology under an electron microscope.
Okay, so it all comes together.
Yes. So if your massive GMP batch tests positive for CD9, 63, and 81, and the NTA proves you have 30 billion particles per mil, and the TEM confirms they all look like little deflated cups, you can confidently say you have a verified, pure, clinical-grade product.
But the source material throws a massive curveball into all of this, specifically regarding the commercial, cosmetic, and personal care industries.
Yes.
What happens when the exosomes aren't coming from human or animal cells? I'm seeing botanical exosome serums and plant-derived preparations popping up everywhere in high-end skincare lines.
Right. Plant-derived extracellular vesicles, or PDEVs.
Yeah.
It is one of the fastest-growing sectors in the industry, but it introduces a fundamental, massive complication for quality control testing.
Because mammalian tetraspanins like CD9, CD63, and CD81 are completely useless here. Like, a plant cell doesn't have human genes.
They absolutely do not. The genetic architecture is completely different.
That is wild to think about. That means applying standard human MISEV marker tests to botanical serums is a massive methodological flaw. If a cosmetic company brags that their apple stem cell exosome serum was verified using CD9 and CD63 markers, they are either lying, their testing methodology is broken, or worst of all, the product is contaminated with human or animal byproducts.
That is the stark reality of the situation. To properly verify plant or yeast-derived exosomes, manufacturers must completely overhaul their quality control panels to test for organism-specific markers.
So what do they look for instead?
Well, if they're making a plant-derived serum, they need to screen for proteins like TET88, which is a plant-specific tetraspanin ortholog, or certain plant aquaporins. If the vesicles are derived from yeast, they have to test for fungal cell wall remodeling enzymes like FKS1.
So for you listening, if you ever splurge on an exosome-based cosmetic or a clinical-grade intimate preparation, the rigorous marker-specific quality control happening behind the scenes, the EXELISA test, the laser tracking NTA, the electron microscopy, that immense scientific effort is the only thing ensuring you are actually buying a functional, revolutionary biological product and not just extremely expensive, purified plant water.
It highlights perfectly why standardized biochemical verification is the absolute bedrock of this entire emerging nanomedicine industry. Without the precise molecular ID badges, we are literally flying blind at the nanoscale.
This has been a genuinely incredible journey. We've traced exosomes from their origins of being dismissed as mere cellular dust in the 1940s, all the way to their current status as the most sophisticated communication network in biology.
That's quite a glow up.
Right. And at the center of it all, we rely on the specific architecture of those tetraspanin markers, CD9, CD63, and CD81, as the ultimate gatekeepers of purity and function.
What's fascinating here is that our ability to verify these complex markers at the molecular level is what finally separates revolutionary science from modern-day snake oil. The engineering required to isolate and confidently identify a 50-nanometer vesicle of fat and protein is a triumph of human ingenuity.
It really is a triumph. But I want to leave you with a final, slightly mind-bending thought to mull over, building on everything we just discussed.
Oh, let's hear it.
We've spent this entire deep dive talking about how CD9, CD63, and CD81 act as these brilliant biological passports. They prove the exosome is legitimate, allowing it to weave through the body and enter target cells completely undetected and unharassed by our body's immune system.
Right. They act as the ultimate biological camouflage.
Exactly. So what happens when bioengineers and synthetic biologists start mass-producing these exact, highly specific tetraspanin markers in a lab? What happens when we take those molecular passports and stitch them onto the outside of completely artificial synthetic nanobots? Could we soon be looking at a future where programmable, man-made machines are moving freely through our bloodstream, bypassing every single immune defense we have, simply because they are wearing stolen biological uniforms?
That is a profound and, frankly, a very imminent reality given the current trajectory of synthetic biology.
Just something to think about the next time you look at a drop of blood or apply a high-tech serum. Thank you for joining us on this deep dive into the source material. Keep questioning the microscopic world around you, because clearly there is a vast invisible universe operating right under our noses.